Oh, great. All right. Let me get my slides up here.
Dr. Graves is new to the ACD community, so we're very excited to have him part of our CCDS conference.
Okay, thank you so much to the organizers for inviting me. As you said, I'm an outsider, and I'm very excited to have this opportunity to talk about our work. Clearly, there's some very interesting research going on in this area, and hopefully maybe something I say in this meeting will contribute to this important mission of understanding how creatine uptake is regulated. So let me just start by introducing my lab briefly. We're a cancer research lab.
So quick mention, we're not seeing your slides, we're seeing your video.
Okay.
So when you do share screen, you probably want to select.
Yeah, sorry. I think I do this when I teach too, and the students are frustrated with me at times. Okay, let's see, here we go. All right, now you should see a slide, and we should be able to say slideshow, play from start. Okay. All right, so can everyone see that now?
Yes, we can, thank you.
Okay, great. Yeah, so as I was saying, our lab is a cancer research lab, and we're primarily interested in trying to understand mechanisms of drug resistance. And to accomplish that, we apply a number of different technologies, largely based on proteomics, studying protein phosphorylation and protein changes, but combining that with metabolomics now to try to understand how metabolism of cells has shifted. Really, like I said, in essence, to understand how drug resistance is occurring. And so one of the cancers we've been studying recently is known as chronic myelogenous leukemia. And this is a very well-defined cancer that's derived from a chromosomal translocation. And what's interesting about CML is that we know it results in the active formation of a kinase known as BCR-ABL, which is a product of the fusion of the BCR protein with the ABL kinase. And because of its well definition, people have been able to design small molecules that are very effective at inhibiting the kinase activity and restoring some of the normal function in patients with this cancer. And so one of the targeted agents you may have heard about is known as Imatinib or Gleevec, and that targets the ABL kinase domain. Now, as one of the first very successful kinase inhibitors, this was shown to inhibit ABL. However, with time, people started to discover that there were mutations in the ABL kinase that prevented this compound from working. And in addition to direct mutations in the kinase itself, there were a number of other mutations in cells that led to resistance. And that's where we got involved, and that was trying to figure out how cells became resistant to imatinib. And our model was a model developed by a Japanese group. And they had taken blood from a patient with chronic myelogenous leukemia, and they continuously exposed it to imatinib until the cells became resistant, as shown in the upper left-hand panel here. So here are the sensitive cells, and here are the resistant cells. What was interesting to us, though, was that these cells became cross-resistant to other drugs, that they weren't dependent on imatinib, but they also resisted other cancer drugs like gemcitabine. And from a variety of studies, we showed that they were not mutated ABL kinase, as I mentioned in the previous slide, and they weren't simple pumps or drug-resistant protein involved in the mechanism of resistance. Instead, we decided to look deeper into what potentially could be contributing to it, and we turned to looking at changes in signaling and metabolism. In the first study that we published on this, we discovered a remarkable increase in the amount of creatine in these drug-resistant cancer cells. So the top panel is the sensitive cells known as the Mil cells, and the bottom panel is the drug-resistant cells known as the Mil-R cells. And by proton NMR, you can see that there was a much larger increase in the amount of creatine accumulating in these drug-resistant cells. And we further showed using phosphorus NMR that a good percentage of it was phosphocreatine, as would be expected. So this sparked our interest because there were other papers that had suggested back in the 80s that there was some increase in phosphocreatine, and some breast cancer models. And we compared across a profile of metabolites, the difference between the MIL and the MIL-R cells. And surprisingly, creatine pool was markedly upregulated compared to all the other compounds that we could measure by the NMR approach. So again, this started us thinking about creatine, which we knew very little about, but we knew that it could potentially be considered important because of its role as a high energy phosphate donor. And shown in the next slide is exactly this, and this molecule doesn't need any introduction to this audience, but we were intrigued by the fact that possibly uptake of creatine, phosphorylation to phosphocreatine, could provide a buffer that might provide some mechanism of survival in these drug-resistant cells or contribute to the development of drug resistance. Now, again, this slide is not necessary for most of the audience, but what we started to consider, well, where is the creatine coming from? Obviously, creatine transport, as we've heard earlier in this talk, is critical to allowing creatine into the cell, where it's rapidly phosphorylated by creatine kinase to form phosphocreatine. And then it provides a variety of functions in terms of cell survival and cell metabolism. The other route is, of course, is the de novo synthesis route, which we've heard from other people, the GAMT and I think AGAT models. So our objectives as a research lab were to really try and understand what the mechanism of creatine accumulation was in these drug-resistant chronic myelogenous leukemia cells. And secondly, to determine if creatine accumulation somehow played a role in the development of drug resistance that we were observing. So the first experiment that we performed was to ask the question, was there a difference in de novo synthesis of creatine in these MIL-R cells? Clearly, we're observing much higher levels of creatine, as I showed. So we asked the question of whether or not this was due to de novo synthesis. And to accomplish that, we used a C13 glycine labeling protocol and proton and NMR to basically measure the increase in creatine. And what we showed was that fractionally, the amount of creatine incorporated, or so the amount of C13 glycine incorporated into creatine from both the MIL and the MIL-R cells was almost identical. That there was no substantial increase in the C13 incorporation in the MIL-R cells, which suggested very strongly that there was no increase in de novo synthesis. So instead, we asked the question, could it be coming from the external milieu? Could it be coming from the media? Obviously, serum is full of creatine. And so we tested this experiment where we tested dialyzed FBS, or fetal bovine serum, and asked the question of whether or not this was due to transport. And as shown in the left panel here, the blue cells are the MIL cells, and you can see normal FBS has a certain level of creatine accumulation. And I should mention that what we did is we washed the cells and then captured the amount of creatine that was inside the cells after these treatments. If we incubated with dialyzed FBS, as shown right here, we saw a decline in the amount of creatine in these cells. Adding creatine back to the media of the dialyzed FBS restored the amount of creatine. And then comparing the MIL-R cells in red, we saw the basal level of creatine was much higher, as we had seen before. Adding dialyzed FBS led to a significant reduction in the amount of creatine. And again, adding creatine back to the media restored it. So this data strongly suggests that it was totally dependent on uptake of creatine from the media. And that was further supported by our data, where we were able to show that a creatine transport inhibitor, 3-GPA, prevented the increase in creatine that we observed in the MIL-R cells. So then we had to ask the question, let's see what's going on here. How is creatine transport regulated? How does this function? And so, not knowing anything about the creatine transporter, we turned to the literature and we started reading about how the creatine transporter works. And what's shown here is, again, a depiction of this complex protein, which again needs no introduction to this audience. Obviously, SLC68 is crucial for creatine uptake, and mutations in this are responsible for many of the creatine deficiencies. But one thing that came to mind to us was that this protein doesn't function alone. And this was reiterated to me by my Russian colleague, Dr. Helmut Hamidov, who said you need an electrochemical gradient for this to occur. Without a proper electrochemical gradient, creatine uptake will not occur. And that electrical chemical gradient is set by the sodium potassium ATPase. And this was an enzyme we were a little bit familiar with because of the importance of this in determining the balance of sodium and potassium in the cells. As in response to the hydrolysis of ATP, it pumps 3 sodiums out and 2 potassiums in. And it's this sodium gradient that's absolutely critical for the uptake of creatine from the media into the cytosol. And this is supported by work from Dr. Silverstein's lab back as far as the 80s, where they showed that either removal of sodium from the extracellular milieu or the addition of sodium potassium ATPase inhibitors prevented the uptake of creatine. And that's, so how does one inhibit the sodium potassium ATPase? Well, I actually teach this in a pharmacology class. This is some very old pharmacology that was pioneered by Dr. William Withering back in the 1700s, where using a mixture of plants, leaves from the Digitalis purpurea plant, we're able to show that these compounds stimulated cardiac function. And the result of this was the discovery of the Digitalis glycosides. Just a second here. The Digitalis glycosides are very potent inhibitors of the sodium potassium ATPase and have been well characterized. And further studies had shown from Dr. Wall's lab that this was important for creatine uptake. And this was published in 1994, but what they showed was both wabane and digoxin strongly inhibited the ability of the cells to uptake creatine, again, reiterating the importance of this sodium electrochemical gradient in driving creatine across the plasma membrane. Now, what was interesting to me about this study was that prior exposure to sodium potassium ATPase inhibitors like wabane or digoxin, when removed, the cells actually had a higher level of creatine uptake. And the authors at the time speculated that there might be an upregulation in the sodium potassium ATPase that was responsible for this, but that was never really formally established. So in our lab, we asked a couple important questions. We first asked, was there any difference in the expression of the creatine transporter in our drug-sensitive or drug-resistant cells? So we compared the MIL and the MIL-R cells. We compared the expression of the creatine transporter by Western blotting. We saw no appreciable difference. And we then also compared creatine kinase. So this data suggested that the levels of this protein were the same. We next tested, would we see inhibition of creatine pools by the Digitalis glycoside wabane? And what's shown here on the right panel is that dose dependently, we saw a very potent inhibition of creatine uptake in the MIL-R cells. And you can see the difference goes from up to 100 nanomolar, which is actually quite a low dose of these compounds. And we showed that similar results using rubidium as a measure of sodium uptake was also blocked by wabane. So that told us that the uptake in the MIL-R cells was dependent on the activity of the sodium potassium ATPase. So we further went back to the literature and said, well, how is the sodium potassium ATPase regulated? And this was where we started to become interested in how post-translational modification and how kinases might play a role in this process. Previous studies had shown that the sodium potassium ATPase could be phosphorylated in response to activation of the insulin receptor. And this seemed to be necessary for activating the function of the sodium potassium ATPase by increasing its pumping action. Other studies had shown that Sarc family kinases could also phosphorylate it, although the site was never identified, and could, in some cases, increase the activity of this. So from previous studies in our lab, we had demonstrated that LIN, a Sarc family kinase, was hyperactivated in Mol cells and was actually required for imatinib resistance. And I won't have time to go into that, but we had established that this was the major Sarc family kinase in the drug-resistant cells. So my graduate student asked the question, could LIN be phosphorylating the sodium-potassium ATPase and upregulating its activity in the Mol cells? And the first experiment that he did was he tested, well, was there more LIN activity, which we had seen before, and using an antibody to this site in the LIN kinase, or the Sarc family kinase, we showed that in the drug-resistant cells that the phosphorylation of the kinase was increased. Importantly, the phosphorylation of P10, or the tyrosine 10 in the sodium-potassium ATPase, was also increased, which suggested that the activity of the sodium-potassium ATPase was increased according to this model. And so we further showed that if we overexpressed LIN kinase in the HEK cells, and we overexpressed an active form, which is shown in lane 3, we saw an increase in phosphorylation of other proteins and an increase in the phosphorylation of the ATPase. So that reiterated this idea that LIN could directly phosphorylate the sodium-potassium ATPase. And conversely, we asked, what about inhibitors of the sodium-potassium ATPase? So we tested a very potent and selective inhibitor known as ponatinib, and we showed that ponatinib reduced the phosphorylation of the LIN kinase and consequently reduced the phosphorylation of PY10 in the ATPase, and also decreased the ability of creatine uptake, or the amount of creatine uptake in the cells. And my graduate student, Dennis, further showed that by knocking LIN down with shRNA, as shown here, also reduced the amount of phosphorylation on the ATPase and reduced the uptake of creatine. So this data strongly supported the idea that LIN phosphorylation was necessary to promote the activity of the ATPase and to promote the uptake of creatine. And so this was our model then, was that LIN kinase could phosphorylate the specific residue in the ATPase, which increased the pumping activity of the ATPase such that sodium would come out and sodium electrochemical gradient would drive creatine in, leading to an increase in creatine in our cells, which then also culminated in the increase of phosphocreatine. So as I said in the beginning, we had sought the question, did this somehow contribute to drug resistance? Well, we were unable to prove that this was actually important for drug resistance, partly because these cells have upregulated other drug resistant mechanisms. But what we did show was that using cyclocreatine, that the Mol cells, the resistant cells, showed a much marked increase in toxicity to cyclocreatine, which was consistent with their ability to take up more cyclocreatine, as shown here. Okay, in summary, what we showed was that there was an enhanced creatine accumulation in these drug-resistant cells that came from extracellular sources. It was independent of de novo synthesis, but was totally dependent on the extracellular milieu, and that the differences in the creatine transporter or the creatine transport level was not due to differences in the creatine transporter per se. Rather, it was an indirect effect on the sodium-potassium, the requirement for the sodium-potassium ATPase, and that was confirmed by the use of the digitalis glycosides. And furthermore, we were able to show that increased LIN activity correlated with increased sodium-potassium ATPase phosphorylation and creatine uptake, whereas inhibition or knockdown of LIN reversed these effects. And so I want to iterate this very last point. I know my time is out, but what our data is showing that the sodium ATPase is very, very important in this function. In this case, it was LIN kinase. But there are other tyrosine kinases, such as the insulin receptor, that can also play a very important role in phosphorylating the ATPase. And that the importance of that could be that modulation of these kinases may ultimately play a role in facilitating creatine uptake, or that this has to be taken into account when considering what strategies one takes to increase creatine uptake. So very briefly, just to say thanks to my lab. This is a small group. The star of the show was Dennis Okumu. He grew up in a small village in Kenya and turned out to be a tremendous scientist and did all the work on the creatine transport with some help